Okay, hi everyone. My name is Natalie Toma, and I'm a current senior at Iolani School. And for my Science Fair Symposium, I'm going to be talking about how heavy metals alter the bacterial communities and pathways in Ulupolo'i. So Ulupolo'i is a native Hawaiian historical site that was once a car landfill during the 80s. And until a few years ago, it was actually still actively growing kalo for the community consumption. However, they discovered that there were extremely high levels of lead in the soil due to car batteries and other waste that was left within the soil. After visiting Ulupo and being surrounded by this incredibly historic atmosphere, it further affirmed my interest in finding restorative methods for this sacred part of our island. Ulupo and many places globally have been severely affected by human activity, which in turn has serious repercussions on our own health. Industrial byproducts like heavy metals are largely responsible for this. Of the 35 identifiable heavy metals, lead is widely recognized as the most pervasive and harmful. This is because lead is a recalcitrant, which allows it to biomagnify as it is consumed by organisms within different trophic levels, therefore disrupting entire ecosystems. In human beings, lead can be absorbed directly into the bloodstream, tissue, and bones, leading to numerous health effects and ultimately death. Every year, it's estimated that 220 million tons of heavy metal pollution are released. Moreover, the World Health Organization approximates that 8.3% of the world mortality rate is attributed to exposure to these heavy metal toxic compounds. The range of lead throughout Hawaii is 0.6 to 380 ppm, with an average of 19.11 ppm, which also served as my study's baseline. The Environmental Health Protection Agency has lead regulatory levels of 400 ppm, and at Ulupo, the highest recorded level is 390 ppm, which is very concerning. So at Ulupo, there were 3 plots that we studied. There was plot A with the highest level of lead, plot B with a medium level of lead, and plot C. And all of the lead was concentrated in the 0 through 10 centimeters of the soil. And if you could look at this picture at the bottom, you can see that plots A and B are very close proximity to each other, whereas plot C is a little bit further down. And this explains a lot of the data. There are a few mainstream removal methods such as reverse osmosis, ion exchange, chemical precipitation, and electrodialysis, but they are very expensive, require a lot of energy, generate toxic sludge while only having partial removal. Another method is known as phytoremediation, which involves using plants to uptake the lead and eventually disposing of these plants. During 2019 to 2020, there were actually phytoremediation experiments conducted at Ulupo using Indian mustard on the left side and ai on the right side. And both plants, as well as the kalo, were analyzed for lead contamination. While there was evidence of lead removal, it was very unsuccessful. In fact, it was estimated to take about 75 years for any significant change. Now, you might be asking, why not just remove the topsoil? And it's because it would be extremely expensive to do so, and it would not be removing the metal particles, simply just displacing it somewhere else. Despite existing methods of heavy metal removal, many aspects of metal-microbe interactions remain largely unexploited in biotechnology. And referring back to the question or the statement I made earlier about not fully removing the metal particles, this is where bioremediation or using bacteria to remediate the heavy metals is very important, because bacteria not only collects these heavy metals within the cell, but it can also transform them into less harmful compounds depending on the pathway. And this is because bacteria actually have a natural tendency to alter and adapt to changing environments. And because Ulupo has been sitting on contaminated soil for about 40 years, the bacteria likely had enough time to acclimate to the soil, which is why I believe it holds great promise in demonstrating resistance to the lead. And I define resistance as some sort of tolerance to the lead, whether it be like absorbing it or transforming it into another compound. Ulupo is a very opportunistic site for microbial diversity because soil just naturally has an abundance of bacteria. And my study looked specifically at the biological pathways, including bioabsorption, bioaccumulation, precipitation, and binding of lead by siderophores. Bioabsorption occurs on the cell surface, and another form of bioabsorption happens when bacteria are under stress. So they secrete a polymer called extracellular polymeric substances, or EPS. And within this film, the lead ions are sequestered. And there's also bioaccumulation, which is when the lead is binded intracellularly, and the metallothioneins are one of the most common proteins that facilitate bioaccumulation. There's also precipitation, but that's less common. The purpose of my project was to determine if the bacterial communities in Ulupo are different, and if the higher levels of lead result in more lead-resistant pathways. I designed my project to find pathways with technologies and databases without having to physically culture bacteria. In 2017, a group of UH researchers collected a total of 30 soil samples. There were 14 in A, 10 in B, and 6 in C. And the soil data was analyzed for both heavy metals and the existing microbial community. The soil data and the 16S sequences created in 2017 were then run through Illumina MySeq. Illumina is a sequencing platform that was used to take the extracted DNA from the sample to determine what bacteria are in the soil. And the result was a FASTQ file, which contained both forward and reverse reads. Then I used QIIME to analyze these FASTQ files and identify the different levels of taxa, such as what genus or family the bacteria belong to. And all the data from QIIME was rarefied, which is a statistical technique that assesses species richness in random subsamples of the community. And because I've had an unequal sampling of the different plots, rarefication was a bias corrector. And then after the taxa data was collected, I analyzed the bacterial pathways. KEGG, or Kyoto Encyclopedia of Genes and Genomes, was used to integrate genomic functional information. Simultaneously, I also gathered a list of bacteria and proteins from other research and UniProt and tabulated how many bacterial families were present under the family name. A microbiome analyst was used to profile and visualize predicted functional genes. And I also created another analysis in QIIME using the 3 samples of the highest levels of lead and the 3 samples with the lowest levels of lead to further draw connections between heavy metal concentration and bacterial behavior and presence. Referring back to the first question of my study, if the bacterial composition was diverse between different land plots, this Bray-Curtis PCOA plot demonstrates that plot A and B are the most similar, while plot C has more distant points. Subsequently, plots A and B have the highest levels of lead. But despite similarities in community composition, each land plot has a very distinctive bacterial presence. If you could look at the first Bray-Curtis plot, you can see that the reds are all clustered together, the blues are clustered together, and the oranges are clustered together. And because of where plot C is, which is orange relative to the rest of the plots, plot C actually has the most bacterial diversity. And it can be suggested that at higher levels of lead, there are less bacterial diversity. And to further connect that point, another QIIME analysis was conducted filtering the highest and lowest levels of lead. And this PCOA plot demonstrates that the soil is habitat filtering, or when the environment is searching for phylogenetically similar organisms. At higher levels, similar organisms are clustered closer together. That is different from that of the lower levels. And it can then be concluded that there is a threshold of lead quantity that is altering the microbial communities. The second question was if the diversity is a direct response to the varying lead levels. It is well established that the toxic effects of heavy metals are highly selective in higher organisms. Invariably, the data indicated a dramatic decrease in genetic diversity with an increasing contamination in the soil. Exposure to soils to higher levels of lead over 150 ppm resulted in substantial community shifts. From plot A, there were levels of lead as high as 390. Majority of the samples from plots B and C had levels of lead that were below 150 ppm and were not altered as much as plot A. Referring to this bar graph, there are varying amounts of different pathways with the highest concentration of families in plot A. The two pathways of greatest interest are the secretion of EPS and the binding of metals by siderophores, or the MTS category, as both pathways have the greatest numbers across plots A and B. Plot A also has the largest concentration of lead-resistant bacteria, which is a direct correlation between the high level of lead in plot A. While the data in the PCA plot indicates that plots A and B are the most similar, plot B has pathways that are more similar to plot C. And metal binding is the pathway that occurs most frequently as form of bioremediation. This table shows 4 different possible lead bioremediation pathways, and these were the bacterial pathways that were found at Ulupiloi. The bacterial families Pseudomonastaceae, Bacillacea, and Enterobacteriaceae were found in every category and have the strongest correlation with bacterial remediation according to other research. Further research should focus on these families to have better results of bacterial remediation. It's also worth noting that every plot had each of these categories, but plot A had more of them, as you can see from the table. This dendrogram demonstrates that there are clustering of similar pathways within land plots A and B, whereas everything else is more distributed. Plots A and B are they're very concentrated within their own area. And this connects back to the PCOA plots, and it expands on the idea that the lead filtered the diversity of the pathways in the bacterial community. Despite not having a very complete analysis of the pathways, I've been able to identify the biosynthesis of siderophore through microbiome analysts, and they were KO1815, KO2552, and KO2361. Siderophores have a very strong affinity for binding metals, and there were only 2 heavy metals that were discovered in the Loei, which were lead and arsenic. But the amount of arsenic that was in the soil were minimal and can be neglected, as the levels of lead do not pass Oahu's mean concentration of 11.92 ppm. So in summary, the levels of lead that were higher than 150 ppm, the communities experienced a large shift, and the microbial diversity was subsequently reduced as a result of these high lead levels to filter bacteria that were specifically suited to lead resistance, and EPS and MTS were the most frequent pathways of resistance found. And through microbiome analysis, I found that the biosynthesis of siderophores were actually present, and they were KO1815, KO2552, and KO2361. As for future research, possible developments in lead bioremediation include further genetic improvement of the previously mentioned biosynthesizing metabolisms. Molecular techniques may permit the design of stains and specific metal binding properties through the expression of metal chelating proteins and peptides, effectively improving the bioabsorption process and introducing metal transformation activities in robust strains of bacteria. Future culturing of bacteria under controlled conditions are also a possibility. Additionally, the soil could be sonified in small portions. Sonification is when you basically vibrate the soil, and what you're left with is whatever is not rocks. And this is under the assumption that the lead is sequestered in the EPS film. In conclusion, bacteria exhibit a number of processes for the uptake and accumulation of heavy metals. The purpose of this project was to discover if any biological pathways are being used to remediate heavy metals and the specific gene functions using 16S RNA data gathered from Ulupoloi. There were several lead-resistant mechanisms that were discovered. Bioremediation of heavy metals within contaminated areas and proper disposal of contaminants can save money, and it is an environmentally friendly alternative. My study has used a direct process for finding functional genes without the aid of culturing bacteria. And by doing so, there was a very cost-effective way of creating biological alternatives to removing toxic metals. Ulupo is not only a place of gathering and food growing, but it is also a place of great historical importance for the native Hawaiian people. As studies within the field of heavy metal bioremediation are still relatively new, this research can be advantageous for native Hawaiian historical sites like Ulupo and ecosystems that have been severely impacted by anthropogenic activities. Being near the end of my high school research journey, I have learned so much about the process of conducting research independently and gained much appreciation for the people working in this field. My advice for younger students would be to genuinely try to search for topics that not only interest you, but topics that you will also be comfortable doing. I know for a fact that I definitely struggled with bioinformatics and using computers. And if I had to do this again, I would probably take a more hands on route. All in all, this was a very informative experience for me. And because of this class, I now have the confidence to conduct meaningful projects that will be beneficial to the community in the future. I'd personally like to thank my mentor, Dr. Prescott, for navigating me through this research project, Dr. Chan and Mrs. Kang for providing me with resources and additional help, Iolani School for funding this research, Mr. Ryan Uenton, who brought me to Ulupo. And thank you so much for coming to my presentation. Let me know if you have any questions.